Have you ever wondered how researchers can identify the specific genes associated with a particular disease? The answer to this is a process called genome-wide association study. Now, hearing this four-letter scientific process might confuse you, but don't worry, I will break this down for you. So let's start from the basics. Every cell in our body has 46 chromosomes, where 23 are from our mom and 23 are from our dad. A chromosome is a thread-like structure found in the nucleus of our cell, and it carries our genes. A gene is a functional hereditary unit made up of a short section of DNA. Our DNA is made up of four nucleotide bases, which are adenine, cytosine, guanine, and thymine. Think of these nucleotide bases as building blocks for DNA. Now think of this according to a computer programming scenario. The code we enter in our computer instructs the computer to perform a certain task. Similarly, our genes also code using these DNA bases to instruct our cell on how to make proteins and further determine our physical characteristics. Lastly, the term genome simply means all the genetic material we inherit, and every cell in our body consists of a copy of our entire genome. Now that we understand the gene and genome, we will better understand how this process works. Researchers primarily conduct genome-wide association studies to identify the genetic variations associated with a disease, or more particular disease-linked trait. First and foremost, researchers will create two groups, the experimental group and the control group. The experimental group will consist of participants with the disease, while the control group will have similar participants without the disease. Next, these participants will provide a blood sample or an oral swab from which each person's DNA is extracted. The extracted DNA is placed on a chip and then scanned using laboratory machines, which can identify all the single nucleotide polymorphisms, known as SNPs. SNPs are genetic variants that are caused due to changes in a single nucleotide base in our DNA. For example, in this picture, the adenine is replaced with the guanine. Upon analysis, if specific SNPs are found to be more consistent in the case group than the control group, they can be said to be associated with the disease. Furthermore, researchers can visualize the location of SNPs in the genome through a Manhattan plot. The x-axis in this graph represents the SNP position in the genome, and the y-axis represents the p-value, while each dot is a single nucleotide polymorphism. The p-value is a measure of the association between the SNP and the chromosome position. The threshold p-value is 5 times 10 to the negative power 18, and if the p-value is less than this threshold value, as seen in the graph or if the SNPs exceed the threshold line, then it can be said that there is a great frequency of SNPs found at this genomic location. However, it is to be noted that genome-wide association studies can only figure out the association between genes and disease traits and cannot definitely state the causation. These studies help health professionals devise a personalized optimal treatment plan according to a patient's genetic makeup. For example, they can now figure out which specific drugs can prove to be beneficial. Our genes play an essential role in disease development. Hence, we must understand the importance of the study and how crucial it is for our future health. Once doctors have analyzed an individual's genomic profile, they can notify the individual of their likelihood of developing a specific disease and what lifestyle choices they should make to prevent it, as most diseases are caused by genetic and environmental factors. So now that you know all about genome sequencing, you can also benefit from this scientific phenomena by going through a genetic test to learn about your ancestry and health risks. Thank you for listening.